everyone, welcome back to NEET Aakash channel. I hope you guys are taking good care of yourselves. This is Lavina Singh, your botany mentor, and today I am going to discuss the questions from slot 2 from your test paper AIATS plus phase 2 mock test 4 that was conducted on 9th of February 2025. Yes, recently you people appeared in this test. I hope you did well, and exam is near now, and I hope you people are taking very good precautions about the the proper things that you have to keep in mind regarding your tips and tricks that you can use in your NEET exam, right? So I am here to discuss the questions from slot 2, and it will cover the chapters biological classification, anatomy of flowering plants, we have plant growth and development, molecular basis of inheritance, and biodiversity and conservation. Alright, so these are there will be the questions that I will be discussing in which you people have made frequent mistakes, okay, so that you don't make those similar mistakes in your NEET exam. For your best preparation, I am here to help in my way, the best way possible. Alright, okay, very good afternoon. Chaliye. So moving to our very first question from this test paper. The question is question number 93. I am covering slot 2. So this is from chrysophytes, that is biological classification. Okay, so the question says which of the following statements is not true for chrysophytes. So what are chrysophytes, in which category do they fall, which are the organisms that are covered under this particular group, also what are the characteristic features. Okay, so such kind of questions you can be asked about a broad term, right, a broad group. You can be given any question, the characteristics will be given, either you have to identify the correct characteristic, or maybe there will be the mention of characteristics in the question, and the option will cover the particular name of the group. Okay, it can be vice versa. Okay, but here we have been given these characteristic features. We have to identify that which one is not true. Pay attention, always pay attention to these not, cannot, right, and odd one out, such terms. Okay, sometimes in hurry we simply take the first answer, oh right, this is the correct one, but the question says which is not true. Okay, so before coming to the options, I want to discuss this group chrysophytes with you. Okay, so chrysophytes fall under the kingdom Protista. Okay, now this kingdom, the most acute characteristic feature of this very kingdom is the organisms are unicellular eukaryotes. Okay, these are the unicellular eukaryotes. Now what we do, we divide this very kingdom into 3 categories. Okay, these categories will be, the first one we see, that is of photosynthetic protists. Okay, so we have the photosynthetic protists. Okay, I can see in the comment section the answer has already been given. We will see whether it is right or wrong, but here I am also discussing the concept related. Okay, so let's thoroughly go through this particular concept so that we can learn better about similar type of questions. Okay, now second we have the consumers. Okay, under these consumers, decomposers, right, and the third category is of protozoans. Now you'll say, ma'am, protozoans are also the consumers only, right, but they are not the decomposers, right, so we have put them in a separate category. All right, now we see that there are certain examples that we can cover under these, like photosynthetic protists, the one that has been given in your question. Example, chrysophytes. Okay, so we have the chrysophytes that will be falling under the photosynthetic protists. Then consumers, or we can simply say the decomposers over here, those will be the slime molds. Okay, and if we speak of protozoans, we can simply mention sporozoans, right? Now we have to focus on chrysophytes. Okay, so let's cover the chrysophytes, right? What are the characteristic features? You already know by now that they are under the kingdom Protista. They are falling under the kingdom Protista. So what are the characteristic features of chrysophytes? Okay, basically chrysophyte is a group, okay, under which the organisms are diatoms and desmids. Okay, for desmids we use another term, that is the golden algae. Okay, now what are the characteristic features? There are certain characteristic features that you have to keep in mind. Questions can be asked about the same. The first one is they are unicellular, right? These are the unicellular ones because it is belonging to the kingdom Protista, and the characteristic feature is those are the unicellular eukaryotes, right? So they are unicellular. They are non-motile, okay? They do not have flagella, okay? So we can simply say they are non-flagellated, okay? But they will have flagella in their reproductive stage, okay? Keep that thing in mind. That is an exception, okay? In their reproductive stage, they will be having flagella, but over here, we simply say they are the non-flagellated organisms, okay? They are non-motile, okay? Then the next characteristic feature is where they are found, the habitat, their home, right? They can be found in fresh water and also marine water. Okay, and they are mostly photosynthetic. They are mostly photosynthetic. Okay, so these are the characteristic features that you have to keep in mind, because the similar question, the question that has been asked in your exam test paper MT04, similar questions can be asked on the chrysophytes. Okay, but most certainly the question can be asked about diatoms, okay, and very repetitively it has been asked in your PYQs also. So I would like to discuss the characteristic features for your diatoms. Over here there is this representation of diatoms, and this is the chrysophyte, sorry, this is the golden algae, that is the desmid. Okay, so can you see the structure? They are the very prominent characteristic feature that we read about diatoms is they are soapbox like structure, right? So I'll write three main characteristics that you have to keep in mind that are often asked in your exam, that is about diatoms. Okay, first of all, these are the chief producers in oceans. Okay, अक्सर आपसे क्वेश्चन पूछा जाता है which are the chief producers in oceans. Okay, so the answer will be diatoms. Okay, so chief producers in oceans. Okay, second very important point is their soapbox like structure. Okay, the cell wall, okay, it is impregnated with silica. ये वाला point I am mentioning, cell wall impregnated with silica. Okay, this question is also asked, this silica deposition, the impregnation of silica, this is also important, impregnated with silica, right? And we have the soapbox like structure. This term, this is there in your NCERT, the questions are asked, okay? Soapbox like structure. Now, why is it so? As I told you, they are unicellular. The cell wall is in such a fashion it is arranged. Okay, we have seen the soap box. There is the lower part in which we keep the soap. There is the lid of that soap box, and when we put them together, they just fit in very well. Okay, similarly, this is the upper cell wall that is called as the epitheca. okay, and this is the lower cell wall, that is hypotheca. Okay, and together they fit in like a soap box. Okay, so these are very important points from your diatoms. Okay, I hope this much is clear with you. Now we will simply move to the question, and the question is about the characteristics of chrysophytes and which is not the true characteristic. The first one says they include diatoms and dinoflagellates. Okay, this is the incorrect one. We are clearly able to see that this is the correct option because this is the incorrect feature, because dinoflagellates are not falling under chrysophytes. Okay, dinoflagellates is a separate group. Okay, so under which we study about goniolux. Okay, so this is the incorrect option. Most of them are photosynthetic. This is the incorrect characteristic and hence the correct choice because it has been asked over here, which is not true. Okay, most of them are photosynthetic. Yes, I just told you about the characteristic feature of them being mostly photosynthetic. They are found in fresh water as well as in marine environment, definitely. And the last one, they float passively in water currents. Yes, they do not have flagella. Okay, and if they are not having flagella, they will be free floating. Okay, and free floating ones are called as the planktons. Okay, the term plankton that we use, it literally means free floating. They are free floating because they do not have flagella. Okay, so this is also a true characteristic for chrysophytes. Okay, but the incorrect one will be the first one because they include diatoms and desmids, that is the golden algae. Okay, I hope this much is clear with everyone. Moving to the next question. Bulliform cells. Okay, this question is from your anatomy. Okay, so bulliform cells are not your anatomy, plant's anatomy obviously. Bulliform cells are present in the leaves of the grasses. Which of the following features are correct? Okay, so we have to see the correct characteristic from the ones that are given from A to E, okay, about these specialized cells. So let us learn about the bulliform cells. Okay, this is also a very important topic, bulliform cells. Okay, let us understand what are the characteristic features that you have to keep in mind. Okay, first of all, these are the living epidermal cells. Okay, the first characteristic feature is they are living epidermal cells. Okay, obviously they are present on the epidermis. Okay, these are the living epidermal cells. Secondly, whenever we are speaking of the bulliform cells, you can see in this representation over here, this I'll mark it for you. These are the bulliform cells. Okay, these are the bulliform cells. You can see that they are occurring in group. Okay, so the second characteristic feature over here is they occur. Okay, they occur in groups. Okay, a single group you can see over here. Next characteristic feature, they are basically large. Okay, they are large in size as compared to the other cells. They are large, they are empty. Okay, they are empty and also colorless. Okay, they are also colorless. Okay, next characteristic feature is basically they are present along the veins. Okay, they are found mostly in grasses. Grasses are monocots, right? That much is clear with you. You can see the structure of veins over here, right? These are the vascular bundles. Okay, now while we are speaking about the vascular bundles, if this vascular bundle is running parallel like that, okay, they are present inside the veins only. Okay, so you can see they are present along the vascular bundles. Okay, so we can say that they are present along the veins. Okay, now why these bulliform cells important? Why they are important? The reason is, let us say this is a grass leaf. Okay, now bulliform cells are present over here. Okay, let us say they are present over here. Now when there is water stress condition. Okay, the plant has to prevent the water loss. Okay, and in order to do that, what these bulliform cells are doing, they are basically becoming flaccid. Obviously they will become flaccid because there is deficiency of water, and when that happens, these cells curl the leaf. Okay, and by curling the leaf, the surface area of leaf reduces, and hence the loss of water can be prevented. Okay, so this is the function of these bulliform cells. So while we are saying that the bulliform cells, they are in the turgid position. Okay, if they are in turgid condition, okay, when they are turgid, they will keep the surface of the leaf flat. Okay, so we can simply say when these are turgid, they will keep the cells or keep the leaf surface exposed. Okay, exposed leaf surface. Okay, because if the cells are turgid, there is no deficiency of water, right? Water is there, the cells are turgid, and hence the leaf doesn't have to curl. Okay, it doesn't have to prevent water. Okay, so in that case, the cells are turgid, the leaf surface is exposed. But if these cells become flaccid, okay, if these cells become flaccid, that means there is deficiency of water. Okay, in that case, they will simply curl the leaf. Okay, the curling of leaf will be observed when the cells are flaccid. Okay, so this is the function of these bulliform cells that you have to keep in mind. I hope this is clear in this particular representation as well. Now, one particular point in which students often get confused is what is the position of these bulliform cells. Now, please pay attention. I showed this particular leaf, grass leaf to you. Now, if this particular leaf has to curl, it curls inwards. Okay, because it has to curl inwards, the position of bulliform cells should be on the upper epidermis. Okay, so they are present on upper epidermis, and upper epidermis means we can use another term, that is the adaxial surface. Okay, keep that in mind. That means the adaxial epidermis. That means the upper part of the leaf. Okay, how I used to keep in mind, I always used to get confused between adaxial and abaxial. Okay, so let's say if this is the leaf, okay, this is the leaf. Now, if I am saying turn the leaf over, what would you do? You will simply turn it like that and you will see the back of the leaf. Usually the shiny part, the glossy part, we consider it as the upper part, right? And the one that is dull, a bit light in color, that is a bit dull, on which the veins and veinlets can be clearly seen, that is the back side of the leaf, right? So abaxial or the back side. In this way, I used to keep that thoroughly in my mind, that this is the abaxial surface or the adaxial surface. Okay, so the adaxial surface is the upper surface because the other one is at the back. Okay, in case of monocots, there is no such differentiation, right? Both the surfaces appear same, but the terminology you can keep in mind to keep this adaxial and abaxial in your head. Okay, so this is the adaxial epidermis where bulliform cells are present. That means the upper epidermis. That is also clear in this representation. Okay, bulliform cells are present towards the upper side. Okay. Very good afternoon, Janak. Very good afternoon, Pankaj. Okay, now moving on to the question. The question asked, which one is the correct characteristic feature? Okay, so first one says, these are modified dead cells of the abaxial epidermis. I just told you it's not abaxial, it is adaxial. Also, they are not dead, they are the living cells. Okay, so these cells are living, first of all right, and they are present towards the adaxial surface. So both of the points that have been mentioned in this particular feature are incorrect. So this is not the true one. Second one says they are responsible to minimize water loss. Absolutely true. They will lead to the curling of leaf and hence prevent water loss. So this is the correct characteristic. Next one says these are large, empty and colorless. Yes, this is the one I discussed with you. Next says, they expose the leaf surface when flaccid. No, when they are flaccid, that means there is deficiency of water. If there is deficiency of water, why would they spread the leaf? They will simply curl the leaf to reduce the surface area, and that is why this is the wrong feature. When they are turgid, leaves curl inward. No, again incorrect, because when they are turgid, the leaf will be exposed. Okay, it will not curl inwards. Okay, so these are the opposite ones. Okay, clear with this? Yes, so the correct ones are B and C, and hence the correct option for the given question will be your first option. Very good, Poonam. Yes, that is absolutely true. Okay, clear with this? I hope bulliform cells, the characteristic features are clear and you will not commit such mistakes. Okay, chaliye. Moving on to the next question, which says, the question is question number 129. It is from the ground tissue system, again anatomy. Okay, anatomy is taking toll on you people, right? You are making frequent mistakes in such concepts. Please sharpen your concepts. Do read your NCERT thoroughly. No matter how many times you have read it again. You will get to know something new every single time. Okay. Hello Govinda, कैसे हैं आप? चलिए, moving on to the next question. Ground tissue system includes, okay, so we have been given the different parts, right, different sections we can say that are present or may not be present in the ground tissue system. So in order to understand, in order to answer this question, we have to switch to the concept, right? So let us understand the ground tissue system with the help of TS of dicot. Tell me in the comment box, this is the TS of what? Which part of the plant? Tell me in the comment box. Okay, so meanwhile, I'll be elaborating this for you, that while we are talking about the tissue system, it has been characterized into 3 categories. Okay, the first category is your epidermal tissue system. Okay. Second one is your ground tissue system that we are gonna discuss just in a while, and the third one is the vascular tissue system. Okay, these are the 3 basic categories we know, right? Now, we see that epidermal tissue system comprises of, first of all, epidermis, obviously. There will be the presence of stomata, as well as the epidermal appendages like hair and trichomes, right? So these are comprising the epidermal tissue system. Then comes the vascular tissue system, because I'll be discussing ground tissue system in detail. Okay, so let's switch to vascular tissue system. What are the members? Xylem, phloem, right? These are our vascular bundles, and we have xylem, phloem, as well as the presence of cambium. Okay, so these are the members for our vascular tissue system. Now moving to the ground tissue system. Okay, so ground tissue system is basically, I will be discussing ground tissue system, all its components, with the help of the TS of dicot. Very good, Hema Malini. The correct answer is dicot stem. This is the TS of the dicot stem. Okay, and it is having all the components of the ground tissue system. In others, we see that some of the components are absent. I will be discussing them also, but in this all are present. That is why I will be explaining the ground tissue system with the help of TS of dicot stem. Okay. So, as we know that just below epidermis, okay, in this particular case of dicot stem, just below epidermis, there is the presence of cortex, okay, and the cortex is also further divided into hypodermis and the general cortex. The first one we have is hypodermis. Okay, while we are speaking of hypodermis, first of all, we should know that what type of cells right? It is comprised of. So in case of dicot stem, the hypodermis is collenchymatous. Your hypodermis is collenchymatous. Tell me in the comment box the type of cells that are found in the monocot stem, hypodermis. Okay, tell me in the comment box. Hypodermis comprises of what type of cells in case of monocot stem? Alright. Next, we see that the cells are collenchymatous. It can be single to several layered. Okay. If we are talking about its function, okay, so it basically is protective in function, okay. So it is protective in nature, protective in function for the plant, and also sometimes it may perform photosynthesis also, okay. Not always, but sometimes it may perform photosynthesis, okay. I am discussing with respect to the dicot stem, okay. Very good, the study empire. The correct answer is sclerenchymatous. If we are speaking of the monocot stem, the hypodermis is sclerenchymatous. But in case of dicot stem, the hypodermis is collenchymatous. Okay. Now, the next layer that we consider is the general cortex. Okay. So, general cortex is present just below the hypodermis. Okay. And if we again consider the type of cells in the general cortex, they are generally parenchymatous. Okay. Secondly, they are basically, the general cortex basically provides the mechanical support to the stem. Okay. So, it provides mechanical support. The next characteristic feature is again, it may perform photosynthesis. Okay? It may perform photosynthesis. So, these are the two sections. I'll tell you in the diagram what we have covered till now, the hypodermis part and the general cortex. Okay, these two we have covered. Now comes the innermost layer of the cortex. I told you the cortex is divided into three parts. Okay, first one is hypodermis, second one is general cortex, and the third one is endodermis. We know endodermis is the innermost part or the innermost layer of cortex. Okay, so I'll clear it and write from the endodermis. Okay. So, next layer is of endodermis. Now, the very important features that you have to keep in mind with respect to endodermis is, first of all, it is single layered, okay. It is made up of, it is single layered. It is made up of barrel shaped cells. What are barrel shaped cells? They are somewhat like this, in the shape of a barrel, okay. So, the wooden barrels are there in which they store grains. Such type of shapes, such type of shape is observed while we are speaking of the endodermal cells. They are endodermis is single layered, made up of barrel shaped cells. These characteristics are very important. The characteristics with respect to endodermis are often asked in your exam, okay. So, barrel shaped cells. They have the deposition of suberin. Suberin is water impermeable substance, okay. So, they are suberized cells, right, and the suberin deposition is in the form of strips that are called as the Casparian strips. So, Casparian strips is another term related to endodermis that you have to keep in mind, okay. This is very important. Casparian strips. Now, this is the innermost layer of the cortex, right? Now, if we are talking about the further, because I'm discussing the ground tissue with you, right? So the ground tissue is comprising of the cortex part, right? And till endodermis, cortex is covered. Next layer that comes is that of pericycle, okay? Yes, okay, so it is pericycle. Now, in case of a dicot stem, the pericycle is present in the form of semi-lunar patches. Okay, over here, can you see these particular structures looking like the pizza slices, right? Now, over here, if we take out one slice, we can simply observe this particular structure. Can you see this red region? In this particular diagram, we are seeing this red region. This red region is in the form of a patch, okay? And while we are looking at the whole structure, they are in the form of semi-lunar patches, okay? So, the patches, first of all, while we are speaking of pericycle, pericycle in case of a dicot stem is basically sclerenchymatous. These are the important characteristic features that you have to keep in mind, because the parenchymatous nature, sclerenchymatous nature, the questions are often asked about them, okay? So please be clear about the types of cells. Sclerenchymatous in the form of semi-lunar patches, okay? Next is, what is the function of the pericycle? Okay, pericycle is responsible for the lateral growth of the roots, okay? Also, it is responsible for the development of vascular cambium during secondary growth, okay? So, what is the function? It helps in the formation of lateral roots and also vascular cambium during secondary growth. Okay, clear with this? Now this is pericycle. The next layer that we have is, okay, the next layer that we have is of pith. That means the innermost layer that we are speaking of, okay? So the centermost region that you are observing over here is pith, right? Now, pith is also called as medulla, okay? This is the another term that we use for pith. In case of dicot stem, we observe that the pith is large, okay? Not like dicot root, it is small and inconspicuous, right? But in this case, the pith is large, okay, and the cells are parenchymatous and they are loosely packed, okay? So, it is comprising of loosely packed parenchymatous cells. You see, in case of dicot stem, we are observing hypodermis, that is collenchymatous, we are observing pericycle, that is sclerenchymatous, and other the region of the cortex or the pith, it is parenchymatous. So all three types of tissues are observed over here, okay? So be thorough about it, be very particular about all of these structures, because when they are related to the other structures like monocot stem or monocot root or maybe leaves, you get confused, okay? So be very particular about these characteristic features, okay? So these are large, loosely packed, parenchymatous. Okay, what is the function of pith? It is basically performing the function of storage. Okay, now we have the last part, that is medullary rays. In this case, we are discussing the whole structure of ground tissue. Okay, so ground tissue is comprising of cortex, pericycle, pith, as well as medullary rays. What are medullary rays? Okay, so I just told you that the pith is also called as medulla. Okay, so the rays arising from medulla are called as medullary rays. You can simply see in this particular representation that between two vascular bundles, we are seeing these rays-like structures that are arising from the pith. Okay, basically they are not actually arising from the pith. The structure is that way that they appear like the rays from the pith. Okay, so we are calling them as the medullary rays. They are also a part of our particular tissue that we are discussing, that is ground tissue. So we have the last one, that is the medullary rays. Okay, and these medullary rays, they are again comprising of parenchymatous cells. Now what is the function of medullary rays? The main function is the lateral conduction of water and minerals. Okay, so we can simply write its function as lateral conduction of water and minerals. Okay, so this is all about the particular ground tissue. Okay, now switching back to the question that we had, ground tissue system includes, okay, so write down the correct, pick the correct answer and tell me in the comment box what should be the correct answer. First one is the medullary rays. Yes, it is a part of the ground tissue. Second one is vascular bundles. No, it is the part of the vascular tissue system obviously. So this is not true. Then comes the pith, I just discussed with you. Then comes the cortex. Yes, it is divided into hypodermis and general cortex and endodermis in case of dicot stem. And the last one is epidermis. But epidermis is the part of epidermal tissue system. Okay, so this is also not true. So the correct answer for the given question should be A, C and D, and that is your second option. Okay, so this is the correct answer. Clear? Okay, chaliye. Now moving on to the, okay, here is a question. The innermost layer of cortex is endodermis. How pith is the part of cortex? Innermost part of cortex, we are talking about the ground tissue system. So the central part of the ground tissue system, we can simply say, right, that is the pith. Okay, the innermost part of the cortex is endodermis. Cortex is finishing over there. After that begins pericycle and then pith. Okay, clear? Yes, very good Govinda, the correct answer is 2. Okay, moving on to the next question. Here we have the simplest expression of arithmetic growth is exemplified by. The question is from plant growth and development. You should be knowing what arithmetic growth is. Okay, to answer it, the concept of arithmetic growth should be clear with all of us. I will be discussing arithmetic as well as geometric growth. Okay, what is the basic difference between the two? So if I am saying that a particular growth pattern is observed and that is arithmetic, what do we understand by that? Okay, let's say there is a cell. Okay, there is, first of all, let's take up arithmetic growth. There is one cell. Okay, the cell divided, okay, and it formed 2 daughter cells. All right, here we see there is the presence of 2 daughter cells. Now, these 2 daughter cells will further divide into 2 daughter cells forming 4, but no, in case of arithmetic, what exactly happens is one cell gets differentiated out of 2 and only one will divide further. Okay, so we can see that such thing will happen that 2 cells will be formed from this very cell. Okay, but the first one was differentiated, that means it became permanent. All right, it was not dividing further. And again the same thing will happen. Now these 2 will become differentiated, right, and only one cell will divide further. Okay, so what will happen again? We will see there will be the presence of these 2 permanent cells. Okay, let's make them differentiated first. I am shading the ones that are differentiated. Okay, and only this cell will divide into 2 daughter cells. Okay, now again out of these 2, one will become differentiated and only this will divide further. So we are observing a constant pattern over here. Okay, only single cell divides rest of them becomes permanent. Okay, so this is how we are understanding arithmetic growth. And if we are considering a graph for arithmetic growth, it will be a straight line graph, okay, because the constant growth is being observed over here, so the straight line graph is over here, okay. And if I want to, question is asked about the related equation or maybe the component of that particular equation, what do we understand by that, okay. So the growth, its meaning, its example and the particular equation, it should be very well clear with you. So the arithmetic growth is represented by the equation, that is LT is equal to L naught plus RT, okay. Over here we see that L represents, let's say, the length, okay, let's say the length. L naught is the initial length, LT is the length after time t, okay. R is representing the rate of growth and T is representing time, okay. So this is the straight line graph. First of all, such sort of growth is observed, only one cell will divide further, rest of them will become differentiated, and the equation for this particular type of growth is this one. Now how do we compare it with geometric growth? What is the difference, okay. So we have the other growth pattern, that is geometric growth. What exactly happens in case of geometric growth? Okay, here on we will observe that if there is one cell, it will divide into two daughter cells, okay. So I have two daughter cells from this. Now both of these daughter cells will further produce four daughter cells. None is becoming differentiated, none is getting permanent, okay. So it will further divide into four daughter cells and these four will further divide to form 8 daughter cells, okay. Clear with everyone? So this is the geometric form of growth. You can see over here the growth, the number is increasing exponentially, right? 1 to 2, 2 to 4, 4 to 8, 8 to 16. This is the exponential growth. So that is why geometric growth is also called as exponential growth. Okay? This is also called as the exponential growth. Now how this particular type of growth will be represented in the form of a graph. So in this case we see if the growth is exponential, right, initially 1 से 2 बने, right, from 1 there is the production of 2 and 4. Initially the growth is very slow, 2 to 4, 4 to 8, but when the power of compounding, that is exactly happening over here, from 16 to 32, from 32 directly 64, the number is rising at a very large scale, okay. So what happens is, initially we will observe that the growth is slow, okay, the growth is slow. Now this is the lag phase, okay, initially this is the lag phase. Now when the number is increasing exponentially, okay, that will lead to a steep rise, right, and there will come a phase that this particular growth will stop, okay, because maybe the resources are limited now, okay. So the initial phase is the lag phase. When the slow growth is observed, then the number is increasing exponentially. The next phase that we will observe over here is the log phase, and then will come the asymptote or the stationary phase, okay. So this particular type of growth, that is the geometric growth, we are observing the lag phase, that means the slow growth, log phase, that means the very exponential growth, and then comes the stationary or the asymptotic phase, okay. So this is the stationary phase, okay. So this is the differentiation हम इसमें observe कर रहे हैं. Now such type of growth, while we are talking about geometric growth, it is observed in initial stages of life. For example, a zygote is formed. Now it has to divide, and it has to divide to form a whole body, right? So initially we will see from zygote, let's say the growth is beginning, okay? So the growth will be exponential, right? Very rapid growth will take place at that time, okay? The body, the particular zygote is not having that much time that the slow growth pattern is observed. It has to divide rapidly to form a mass, okay? So that time the growth is, this is just an example, okay? So that time the growth will be exponential, okay? But whenever you observe that, can you see this particular straight line, right? This is representing the constant rate, right? Whenever something is growing at a constant rate, at that time arithmetic growth is observed, okay? Clear with this? Now moving to the question, what it asked the simplest expression of arithmetic growth is exemplified by, first, all cells of a plant. No, all cells of a plant cannot show that, okay? Some cells will begin to differentiate, right? For example, in root, we observe there is the meristematic region. After that, from the meristematic cells will become differentiated, right? And they will enter the elongation phase, and then those particular cells, first of all, they will enter the elongation phase, and then they will differentiate to form the maturation phase, right? So basically, we cannot say that all cells are growing through this particular arithmetic rate, okay? Some are growing at the geometric growth pattern, and some are taking up the arithmetic growth pattern. So this is not true. Bacterial cell in a culture medium, yes. Arithmetic growth, if we are talking about arithmetic growth in bacterial cells, the growth will be geometric, right? Because we know from one bacterium, there will be formation of 2 daughter bacteria, and further 4 to 8, 8 to 16. Again, the growth is exponential in that case, okay? So this is also not true. Then root elongating at a constant rate. Always remember, if something is growing at a constant rate, that means it is taking up the arithmetic pattern, okay? The last one says initial division of a zygote. As I told you, it is not true. The growth is geometrical in that case, okay? Clear with this? I hope. Now moving on to our next question. The question says, okay, it is from your molecular basis of inheritance. Study the following statements from A to D given below, okay? And we have to pick up the incorrect statements. Let us see what the concept is about. The code is read in mRNA in a contiguous fashion, okay? So basically we have been given the characteristics of a genetic code, okay? So let us first discuss all the salient features of the genetic code, okay? So while we are speaking of the salient features that are there in your NCERT, questions are often asked, they are very, very important, okay? So salient features of genetic code. What is genetic code? What do we understand by genetic code? Basically, all the amino acids are having a particular code, okay, of nitrogen bases, okay, of the nucleotides, okay, we have, and those particular nitrogenous bases will be representing a particular amino acid, and they are present in the form of triplets, okay. For example, if I say about AUG, okay, so these 3 alphabets are representing the particular nitrogen bases, adenine, uracil and guanine, okay. Now this will lead to, this is giving to the production, this is a code for a particular amino acid, that is methionine, okay. So if this particular methionine, okay, if this particular code is present, right, that means the production of methionine amino acid will take place, okay. This is what we understand by a code for a particular amino acid, okay. Yes, very good, Tanmay. The codes are used for coding the amino acids. So what are the salient features? The first salient feature is they are present in the form of triplets, okay. So 3 alphabets will be written, okay. Alphabets as in not any random alphabet. We have adenine, guanine, uracil, thymine, right, cytosine. These are the ones, the nitrogen bases, okay. So the nitrogen bases will be present in the form of a triplet, okay. So for example, GUG, right, UUU, or you can say UUC, these are the particular codes for particular amino acids. Second one, the code is basically comma-less. What do we understand by comma-less characteristic? That means while we are writing a whole genetic code, it is not in the form of breaks. It is not in the form of the fragments. Okay, it is right in continuous manner. Okay, so the genetic code is read in a contiguous fashion or contiguous manner, we can also say. Okay, so it is read in a contiguous manner. If I am writing a genetic code, I will not write it as AUG, GUG, GUC, GUG or whatsoever. Okay, I will not put the comma over there. Okay, it is read in a contiguous fashion. Okay, so it is comma-less. Second, the third characteristic feature over here is the genetic code is basically degenerate. Okay, now this is very important characteristic feature. Question is asked, what do we understand by the degeneracy of the genetic code? It says that one amino acid can be coded by more than one code. Okay, what do we understand by this? Let us say I have UUU, okay, and I have UUC. Okay, both are going to code for phenylalanine. Only one amino acid I am speaking of, but the codes are two. That is UUU and UUC in this particular example that I am taking. Okay, so both of them are coding for a single amino acid, that is phenylalanine in this case. Okay, so this is what we understand by the code being degenerate. Okay, now there comes an exception for the same. Okay, not all the amino acids are having multiple codons for them. Okay, not all of them are having multiple codons. For example, we have AUG. Okay, AUG is only coding for methionine. And methionine is not being coded by any other particular codon. Okay, in this case, phenylalanine is being coded by UUU and UUC, both. But if I am taking the example of methionine, or let us say tryptophan, these are basically non-degenerate. Okay? Degenerate wala फिर से बता दूं. Okay, Xenia, pay attention. If I am saying a code is degenerate, that means a particular amino acid is being coded by more than one codon. Okay, over here phenylalanine is here. Phenylalanine is coded by this code also and this code also. Okay, there can be multiple codes, right? But if I am saying that the code is non-degenerate, the exception is there. Okay, in that case, I am taking the example of methionine. Methionine do not have multiple codons for it. It is only having a single codon, that is AUG. Okay, it will not be coded by any other else. Okay, clear with this? Now, moving on to the next characteristic feature, that the genetic code is basically, see, these three are very important. ये आपको अच्छे से याद होने चाहिए. Okay, even all the characteristic features you have to keep in mind. Okay, do not forget them. Now, next feature is the genetic code is nearly universal. Why not universal? Why nearly universal? Let us understand. This word nearly is very important for that, because while we are talking about a particular code, okay, let us say in my body, UUU is coding for phenylalanine. It will code for phenylalanine in case of a dog also, in a bacteria also, in elephant also, everywhere it will be coding for phenylalanine. Okay, that is the universal nature of a codon. Okay, but if I am saying that it is nearly universal, that means there are some exceptions. Okay, so focus on nearly and follow the exception. The exception is in case of mitochondrion. Okay, अगर हम बात कर रहे हैं about a genetic code, let us say UGA. Okay, in my body, right, in normally it is coding for methionine. Okay, normally it is coding for methionine. Okay, I'm so sorry. It is coding for tryptophan. Okay. If I'm saying in my body, it is, okay, if it is UGA, it is a stop codon, right? And stop codon means it will not code for anything. But I told you, in case of mitochondria, it is coding for methionine. Now, this is the nearly universal nature, right? In mitochondria, it is coding for methionine, but otherwise, UGA is a stop codon. That means it doesn't code for anything, okay? So, this is the nearly universal nature of the particular genetic code, okay? Okay, we have only, no, we have 10 amino acid. Why the need of 61 codon is there like for 20 amino acid? See, this is the reason, this degeneracy is the reason behind it. One particular amino acid is not coded by single particular codon, okay? It can have multiple codons for that, right? So, for 20 amino acids, we are having total 64, right? But out of those 64, only 61 code for the amino acids and the rest 3 are, okay, over here I will mention two important ones, that is the stop codon and the start codon, okay? So, we have one start codon and this start codon is having dual functions, okay? That is AUG. It acts as the start codon, but it is having the dual function. It also codes for methionine, okay? Also codes for methionine. Okay, so it is serving dual functions. Secondly, that means 61 codons are still there, but the rest 3 codons that we are speaking of, they are called as the stop codons. Now, these stop codons do not code for anything except in mitochondria. I just mentioned the exception for that. UAA, UGA, and UAG. Okay, these 3 codons are also called as the nonsense codons, okay, and they are wherever they are present on the mRNA. Let us say there is the whole strand of mRNA and there occurs this particular codon, let's say UAA. The process of translation will stop right there because it is supposed to stop the particular process, okay. So, 64 codons are there, right? We have been given a table in our NCERT, but out of them only 61 code for amino acid and the rest 3 are the stop codons, okay? Clear with this? I hope this is clear and we will be answering these. The study the following statement. The code is read in mRNA in a contiguous fashion. That is absolutely true. The base sequence for particular amino acids are always same for a particular species. Okay. So if we are taking up this particular point, the base sequence for particular amino acids are always same. No, it is not always same. Okay. So this is not true. In 64 codons, 4 codons do not code for any amino acid. No, only 3 codons are there that do not code for any amino acid. So this is also not true. Some amino acids are coded by more than one codon. That is the degeneracy. That is also true. So the correct statements are A and D and the correct answer is your second answer. Okay, clear with this? Any doubt, anyone, please tell me in the comment box. I hope this much is clear with you. You people made a lot of mistakes. Many of you made this particular, committed mistakes in this particular question. So please do go through your NCERT thoroughly. Read all the particular points that have been mentioned regarding the genetic code over there. Okay, and be prepared for such kind of questions. Alright, now the last question that we have is about 1,000 varieties of mango are found in India. This is an example of, okay, so 1,000 varieties of mango are there. See, while we are bifurcating the particular, diversity, biological diversity, it can be species diversity, it can be ecological diversity, also called as community diversity, or it can be the genetic diversity. Okay, so we have 3. Okay, so we have genetic diversity, we have the particular species diversity, right? And the third one is the ecological diversity, right? While we are speaking of the genetic diversity, we have the example of Rauwolfia vomitoria. Okay, Rauwolfia vomitoria is a particular plant that is found in the Himalayan ranges. Okay, now in Himalayan ranges, let us say this is the whole range. Okay, let us say this is the whole range, and one Rauwolfia is growing over here. Okay, and the other Rauwolfia vomitoria is growing over here. Okay, both are Rauwolfia vomitoria, both are the same species, but in this particular plant, the amount of reserpine, okay, that is potency of reserpine that is found in this particular plant is less, whereas in this particular case it is more. Reserpine is a chemical that is derived from the roots of Rauwolfia vomitoria, okay, that is used to make medicines for high blood pressure. Okay, so this particular plant, same species, but they are having genetic diversity, and because of that genetic diversity, same species is producing two types of variants. Okay, so this is the example of genetic diversity over here. So next comes the species diversity. The next species is Rauwolfia serpentina. Okay, it also produces reserpine, but over here the species are different. One is Rauwolfia vomitoria, other one is Rauwolfia serpentina. Vomitoria and serpentina, two different species. Now that comes under species diversity. Okay, and the last one comes is the ecological diversity. What do we understand by this? When there is diversity of ecosystem. Okay, the example that has been given in your NCERT is of Western Ghats and Eastern Ghats, the number of amphibian species. Okay, so this is the this particular region is the Western Ghats of India, this one is the Eastern Ghats of India, right? But if we are considering the Western Ghats, the amphibian species are much more in number over here, okay, as compared to the Eastern Ghats. Okay, so this is called as the ecological diversity. Okay, the number of the variety of ecosystems that are found, okay, that is called as the ecological diversity. I hope this is clear with you people. Now the question was asking, ecological diversity is also called as the community diversity. So the correct answer for the given question, now variety of mango have been asked. No matter what variety of mango is there, whether it is Dashehri, it is Tota, Langda, whatsoever mango is there, the species will remain the same, right? That is Mangifera indica. Okay, so but there are 1000 varieties, so the correct answer is genetic diversity. Only the genetic level changes are taking place, that is why so many varieties, but the species is still remaining the same. So no species diversity, not ecological, neither this particular community diversity. All right, I hope this explanation is clear with you. You will not commit those errors again, and the next slot is being taken by our next faculty, that is Aman Vishwakarma sir. He will be taking up the questions from rest of the chapters, so stay tuned.